Hello friends, welcome back to my YouTube channel Nesting Padasala. In this video, we'll be discussing about the question that has been asked for this DBT Biotech Industrial Training Program. First question is, primary carboxylation takes place with the help of which enzyme in C3 plan and which enzyme in C4 plan. In C3 plan, it is taken over by the ribulose bisphosphate carboxylation. In short form, it is called Strobisco. And in C4 plan, it is carried over by phosphoenol pyruvate carboxylase. So which one of the following is the correct electronic configuration of fluoride atom? Fluorine atom, it is 1s2, 2s2, 2p5. Option A is correct. Rancidity in spoiled food is due to oxidation of unsaturated fatty acid. Next is a bioinformatics question. What is the computation methodology that is used to predict the potential binding between a receptor and its ligand? It is molecular docking. Which one of the following carboxylic acid is a chiral? So the correct answer for this question is lactic acid. Next question is regarding an analytical kind of question. It is asked from blue-white screening technique. Okay. So while cloning, you attempted to insert a gene of interest into LAC-Z gene in a vector that has an ampicillin resistant marker. After ligation, you had transformed and plated the cells on X-call plate lacking ampicillin and obtained a large number of colonies and most of them are white. What is the most plausible explanation? The correct answer is most of the colonies are from cells that have not been transformed by the plasmid. Okay, so in the next video I'll be explaining you about this question number 6, which many people you are confused about this particular question number 6. So we'll be discussing that in the next video. Identify the correct answer for how do touch screens work on your smart mobile phone. So it works by using a finger to disrupt the electric current. Next question is, specific cleavage at phenylalanine, tyrosine, tryptophan amino acid is catalyzed by. It is catalyzed by chymotrypsin, which will be cleaving the peptide bond that are present between an aromatic amino acid, since this phenylalanine, tyrosine, tryptophan are coming in an aromatic amino acid category. Next, which one of the following bond is more stable? The correct answer is disulfide bond. Disulfide bond is a kind of covalent bond. It will be formed between two sulfur atoms. So correct answer for this question is disulfide bond. Next is, which of the following statement is false about the mammalian mitotic cell cycle? The correct answer is option B. Here they just mentioned cyclin A peaks during G1 phase. But actually, here if you watch, cyclin A will be peaks during synthesis phase, and in G phase, cyclin D will be higher amount. Okay, cyclin D will be taking and regulating all the aspects of the G1 phase. Next is, Rohit performed quantitative real-time PCR for estimating the expression of gene of interest in two samples A and B. He observed similar CT value for a housekeeping gene in both samples. If you find that the expression of gene of interest is higher in sample B, then ultimately CT value of gene of interest will be lower. So if expression of gene of interest is higher means then CT value will be lower. If expression of gene of interest is lower means CT value will be higher. They are inversely proportional. Next is A, B, Z DNA respectively. So A DNA and B DNA, they have a left-handed rotation, and Z DNA they have A DNA and B DNA, they have a right-handed rotation, and Z DNA will be having a left-handed rotation. Next is, lethal dose of a drug required to kill 50% of lab animal in a test group and a standard condition is defined as. So it is defined as LD50, L for lethal, D for dose. Okay. Whereas IC50 means half maximal inhibitory concentration. So it is generally used in pharmaceutical field. And IC50 will be indicating how much a drug is needed to inhibit a biological process by half. So it is generally providing a measurement about efficacy or potency of a particular drug. Next, there is a question about solvent flux across the membrane. So the correct answer is option A. Next is, upon infection with the necrotizing pathogen, many plants will be developing an enhanced resistance to further pathogen attack also in their uninoculated organs. This type of enhanced resistance is referred as systemic acute resistance, which will be providing immunity in all plants. Just like an immune system in humans, plant will be having the systemic acute resistance, and it is mediated by jasmonic acid, whereas some cases it also be mediated by salicylic acid. So both the jasmonic acid and salicylic acid, they are involving in plant defense mechanism. Next is, identify the odd one out of the following set of number. The correct answer for this question is 27, but according to me, 23 is the correct answer. So if you have some other answer, or if you are stating 27 is the correct answer, please do tell me how you are stating 27 is the correct answer. Okay, I don't know how to solve that number, that's the reason. So next question, if the concentration of ligand is equal to Kd, then the fraction of ligand bound to protein will be, ultimately if concentration of ligand is equal to Kd mean, then ultimately what will be there? Fraction of ligand bound to protein will be half. So 0.5 is the correct answer. Next, which isotopes or which atoms are generally incorporated into protein for use in NMR study? So most commonly used isotopes that are used for detecting NMR signal include H1, C13, but there are many other isotopes of hydrogen like deuterium, helium-3, nitrogen-15, fluoride-19. Okay, so nitrogen, fluoride, helium, hydrogen, carbon are generally used, and the isotopes of those atoms are generally employed for detecting the signal in NMR study. So correct answer for this question is nitrogen. Next in agrogel electrophoresis, which of the following will move fast? So supercoiled DNA will be moving fast. So in this diagram, you can able to see first supercoil will be running, followed by neck DNA and followed by linear DNA. So supercoil, they have a compact structure, so they can easily move through the agrogel electrophoresis. Okay. Next, as per the patent act, how often a patent need to be renewed for maintaining the protection? Okay. So here they are asking a question about renewal. So many students had given option C, that is every 20 years is the correct option. But every 20 years is the term of patent. But here they are asking every year. So renewal about patent, if you are going for applying for patent means once they had allotted a patent ID number means then you need to renew a patent every year by paying amount of particular fees. Okay, for the first 2 years there is no need to pay any amount. Whereas starting from third year you need to renew a patent by paying amount of particular fees, or else they will be seizing your patent. Next, what is the approximate size of human genome? It is 3 billion base pair. Next, how many different type of gametes are produced by heterogametic females? So, in some organisms like fishes, insects and birds, females are heterogametic and males are homogametic. Okay, if you take humans, in humans, males are heterogametic and females are homogametic. Okay, so what is mean by heterogametic means those organisms or those animals will be having a two different sex hormone. So ultimately, two different types of gametes will be produced. So the correct answer is 2. Next is which one of the following DNA duplex will have a highest TM value. So for this question, I don't know how many of those challenge this question, because in answer key they just mentioned DNA duplex with 16% of AT content is the correct answer. But according to me, option D, DNA duplex with 48% of GC content is the correct answer, because they are asking which of the following duplex will be having a highest TM value or melting temperature. So those DNA sequences which are having a more number of GC content will be having a highest TM value. Next, what is the generation time if 100 bacterial cells? So initially there were 100 bacterial cells growing logarithmically for a period of 1 year. So time is 60 minutes, that is 1 hour. Produce a final bacteria is 10,000 cells are there in the final. So generation time they are asking. So generation time formula is T divided by N. T we know it is 60 minutes. What N is the number of generation. This is the formula of number of generation, that is 3.3 log of N divided by N naught. So 3.3 multiplied by log of 10,000 divided by 100. So N is 6.6, and now if you calculate generation time, it is 60 divided by 6.6. So our correct answer is 9.09. Okay. Next is an analytical kind of questions. You are using an automatic pipette that measures about 1 to 20 microliter. You are told that particular pipette is faulty and measures 20% more than the set volume. Now the question is that to correctly measure 6 microliter, what would be the set value in microliter? Okay, so the correct answer is 5. If you set 5 microliter in the pipette, then it will be correctly measuring 6 microliter, because the pipette is with the mistake, there is something fault and measure 20% more than that of the set volume. So correct answer is 5. Next, which of the following is the correct conversion of 1 calorie in term of joules? Okay, so this correct answer is 4.2. 1 calorie is equal to 4 joules. Next is in a class consisting of 60 students. So total student in the class is 60. There are twice the number of girls than that of boys. So totally 20 boys are there and 40 girls are there. And Narayan obtained 8th rank. So before Narayan, there are 7 persons getting first rank, second rank to 7th rank. Okay, there are 3 girls above him on the rank. So total number of students is 3 students are girls, and so ultimately remaining 4 will be boys. Okay, so the question is that identify which of the following is the correct number of boys below Narayan. Okay, so they are asking only boys. So totally 20 boys were there in the classroom. So 20 minus 4 minus 1. So minus 1 is for, we are counting Narayan, which is who is also a boy. So correct answer is 50. Next, which of the following pairs could form a hydrogen bond in solution? The correct answer is water and ethanol, because they have a OH group. Rest of them are hydrocarbon compound. They don't have a capacity to bond to form the hydrogen bond. So correct answer is water and ethanol. Next, which of the following microorganism oxidizes nitrites to nitrate? The correct answer is nitrobacter. This nitromonas is involved in converting your ammonia into nitrates, and the acetobacter is called as free nitrogen fixing bacteria, where they will be fixing the atmospheric nitrogen in the form of ammonia. Next, expression vector is used for expressing a protein, and a simple cloning vector is for cloning a gene. Differs in which of the property? Okay, so expression vector and cloning vector, they are having a simple skeleton only, but only difference is that in expression vector you can be able to find a strong promoter for the gene of interest. Additionally, expression vector will be having an enhancer sequence, they will be having a transcription start set, some other sequence. Okay, in both expression vector and cloning vector, you can be able to find the origin of replication will be there, there will be a selection marker will be there, and there are also multiple cloning sites will also be there. Only difference in expression vector, there will be a presence of strong promoter will be there. The headquarters of Indian Patent Office is located in Kolkata. I don't know how many of them had clicked New Delhi. The correct answer is Kolkata only. What is Agent Orange? So Agent Orange is a herbicide containing dioxin chemical group. So Agent Orange was initially used long back by US military in order to clear few vegetation in the forest at time of war. But in the year 1971 itself, this Agent Orange has been banned in many countries as it is reported to cause cancer in humans. Okay. Next, what is the principle behind 3D visualization under stereo zoom microscope? So the correct answer is, in stereo zoom microscope, there will be two light paths with an angular deviation. So that's the reason we can able to see a 3D visualization. Next is streptolysin O is inactivated by. So streptolysin is endotoxin which is produced by Streptococcus bacteria, and they will be producing two exotoxins. Sorry, it is an exotoxin. So streptolysin O and streptolysin S. So streptolysin O is inactivated by oxygen, whereas if you take streptolysin S, it is not been inactivated by oxygen. Okay, so streptolysin O is alone inactivated by oxygen. The correct answer is oxygen. Which of the following statement is not true regarding the law of segregation? The correct answer is, allele frequency and genotype frequency in a population will remain constant from the generation to generation. Okay, so this is not true. The correct answer is B. Which of the following is inactivated virus-based vaccine against COVID? Okay, the correct answer is Covaxin, which is produced by Bharat Biotech. It is the first vaccine and it is an inactive virus-based vaccine. Next, which of the following recombinant DNA-based product is developed for treating hemophilia? The correct answer is coagulation factor 9 is employed for treating hemophilia. Okay. Next is flow cytometry. The forward scatter gives the estimation of. So forward scatter will be giving the estimation of cell size, whereas side scatter will be giving an estimation of cell granularity and cell density. Next, transplantation of tissue between identical twins is called as isograft, whereas autograft means skin obtained from the patient's own donor cell. And allograft means skin will be obtained by some other person. And in xenograft, skin tissue will be obtained from some other organism. Okay. Next was a question regarding to FT infrared spectroscopy. The mathematical model known as FT is used to transform signal. It is used to transform the signal from the frequency domain to the time domain. Next, which one of the following metal ions are utilized in this reaction? The Mg ions are generally used in this particular reaction. Next, which one of the following has a higher photon energy compared to ultraviolet rate in the electromagnetic spectrum? The correct answer is X-ray. X-ray are having a high photon energy when you compare them with the UV rate in electromagnetic spectrum. Next, a spontaneous point mutation generating a variation in pathogen to create a new strain is referred as. So, it is referred as antigenic drift, which is a minor strain within a subtype. It is caused due to point mutation. Okay, so once this particular antigenic drift occurs, then there will be occurrence of two different subtype of a particular virus. Okay, so this antigenic drift is responsible for causing epidemics, whereas antigenic shift, it is responsible for causing pandemic, and it is due to exchange of genetic segment. Okay, the correct answer is antigenic drift. Next, which of the following segment are associated with the process of photorespiration in plant? Okay, so the correct answer is option number C. I think many of you will be giving a correct answer. So glycolate is oxidized to glyoxylate in peroxisome. So in peroxisome only, the glycolate will be oxidized to glyoxylate. Next is which one of the following is not autoimmune disorder. So you should read the question correctly. They are asking which is not autoimmune disorder. The correct answer is type 2 diabetes. Whereas type 1 is a insulin dependent diabetes where the self antigen will be present in the pancreatic beta cell. And Hashimoto thyroiditis, self antigen will be present in the thyroid cells and thyroid protein. And myasthenia gravis, ultimately the self antigen will be present in the acetylcholine receptor. Next, which kinase activity is associated with the phytochrome photoreceptor responsible for red and fair light response in plant? It is histidine kinase activity is associated with the phytochrome photoreceptor responsible for red and fair light response in plant. Next, which of the enzyme is primarily responsible for the movement of transposomal element? Okay, mobile genetic element, the other name is transposon. So the correct enzyme is the transposase enzyme is involved in moving the mobile genetic element from one place of gene to another place of the DNA. Okay, so which of the following experimental method is not used to determine the three-dimensional structure of biological macromolecule? Again, it is a repeated question. You can able to find this question in DBT examination. You can able to find this question in GATE, Excel, BT, and even CSR examination. The correct answer is CD. It is generally employed for determining the two-dimensional structure of protein, whereas test of NMR, X-ray, electron microscopy are employed for 3D structure. Next is hybridoma technology. One question has been asked. In that, during the production of hybridoma cells, amnopterin is used to. It is generally used to block the salvage pathway. Amnopterin is generally used to blocking the salvage pathway. This is also a repeated question. Next, which of the following structure mentioned below is found in the cytoplasmic compartment? So, correct answer is stress granules. So, these stress granules are produced when at the time of translation, few mRNA will be stalled at the initiation site itself due to few kind of stress to the DNA. Okay, so this stress granules will be present in the cytosol environment, whereas rest of the thing like PML bodies, Kajal bodies, paraspectral, they are found in the region of nucleolus, whereas the Kajal bodies will be found in the nucleolus portion. Okay, next is dicer. So dicer is an endonuclease. Next is, which of the following is a multiple sequence alignment tool? The correct answer is cluster W. BLAST is a pairwise alignment sequence tool, and PRINCE. So PRINCE is a database for determining the protein family, and OMIM, online Mendelian. So it is a database where you can be able to find the human genes responsible for causing few genetic disorders. So it is a software bioinformatics database. Okay. Next, so we'll be seeing the rest of the question in part 2 of the video. Thank you friends. Thanks for watching this video.